In this tutorial, I am going to show you how you can generate heat maps using R. So by the end of this tutorial, you'll be able to generate this heat map that is being shown right now. Let me also say that the code for this tutorial, all the codes have been packaged into an R script. So if you want that R script, then check the description box. The download link is there. In order for this tutorial, you need to make sure that you have these two packages installed. These two are packages. So this one and then this one. So this one here, the gplots, okay, this will be used to generate the heat maps. And then the R color brewer, this will be used to set the colors. If you have this one, it's fine. This is the main library we'll use, but I'll advise that you install this one also so that you can set the color to whatever you want. Now for this tutorial, we are going to reproduce this online Galaxy tutorial. Let's just visit that page. So we are going to reproduce this online Galaxy tutorial here. I'll leave the link to this page in the description box. So this tutorial shows how to visualize RNA Seq results with Heatmap 2. So we are going to reproduce this tutorial using our local R environments. And by the way, I'm going to make another tutorial where I show how to use the Galaxy platform to also generate the heat map. So make sure to come back to this channel and then watch that. Now let's proceed. So I also encourage you to read the introduction here, especially for a beginner. Heat maps are used to visualize RNA Seq results. So they can be used to identify groups of genes with similar expression patterns. They can also be used to identify signatures associated with a particular condition or conditions. So just take note of that. So that's just a brief description of what heat maps can be used for. But read the introduction here, is going to help you. So let's go down. So for this tutorial, we need the inputs. So when we are here, come to preparing the inputs. So click it and you'll be sent to this section here. So these are the input files. So we have normalized count files, and we also have differential express result file, and we also have the genes of interest. These are the three files we will use. So scroll down and we are going to find the download link. So these are the files. So we have the normalized count file, the differential expression analysis result file, and then the genes of interest, which can be found in this file. So make sure to download them. So to download, just highlight the link, this one for example, and then open it. So I'll right click, then I'll go to this one here, go to this link. I am using a Google Chrome, so that is why I have this option. But if you are using Firefox, then instead of go to, you may see open link or something like that, and then you have to click it. Or you can also copy and then paste in a new tab, but I will just use this option here to download. So this one has been downloaded for me. You can do same for these ones. So try to do same for the other two and get all the files. I already have mine downloaded, so let's take a peek into the files. So I have the genes of interest, I have the differential expression results file, now I have the normalized counts. So I think I will start with this one first, the normalized count. So if you download, it's likely you may observe that these files have no extension. You know, sometimes you have file.csv or .tsv or .xls, etc. So these ones, they do not have extensions. But you can open them with spreadsheet softwares, or you can also open them with text editor. But these are that are tab separated. So let me just do it this way. So what you want to do is to open with your spreadsheet softwares. So I'll start with a normalized count. So I'll just open mine here. I'm using my spreadsheet software. So there's it. So if you're asked about the separator, select the tab separator. So this is the normalized count. So we have the conditions here, and then we have the information about the genes. So we have the ID symbol, gene name, and then we have the conditions here. So each row represents a gene, and then for each gene its count has been recorded for each of the conditions here. That's what you have. Let me just zoom in, make it bigger. Yeah, this one should be fine. So this gene, for example, there's the counts in this condition, etc. So these are normalized counts. We also have another condition. So these are normalized counts. So you need to make sure that you get this far. By the way, I have a tutorial that shows how to generate read counts. So you can check the description box for that video as well. So that's what we have here. So every gen and then its respective count in the different conditions. So we have the conditions here. Anyway, we will do some exploration in the R environment as well. So that's about it. So that's for the normalized count. Let's take a look at this one here. This is the differential expression results file. We can also open it with a spreadsheet software. So that is it. Yeah, we can enlarge it a bit. So for every gen, we have the log 2-fold change. We also have this average expression, p-value adjusted, and all those ones. We also have this one, etc. So this is the differential expression analysis result. So you can use tools like the Seq2, HR, Borgon to generate this kind of data. So I've covered some tutorials on that, by the way. So check the description box, you'll find links to those videos. So that's about it. And we also have the Heatmap gens. This one, okay, let's just open with a spreadsheet software. I think this is just a single column. Yeah, so we have the gens of interest. So what we are going to do is that we are going to generate heat maps. Let's go back to the Galactic tutorial. We are going to generate heat maps for the top DE gens. So D is the differentially expressed gens. So the top DE gens, we are going to generate a heat map for that, which is this one. And then we are also going to create custom heat map. That's for this one here. That's what we are going to do. So basically, these are the two heat maps we are going to generate. One for the top D gens, and then the other one for the custom gens. We will take a look at all of them. Perfect. So that's about it for the intro. So let's start the exercise. So the instructions, we need to take a look at them. So we will start with this one here. It says rename the count datasets as normalized counts, and then we should also rename the lemma vom etc. This long file, we should rename it as D results. That's what we should do. So we are basically going to follow the instructions there, because the names are long and it can be complicated. So let's start with the normalized count. So mine is here. I'll just do it this way. I'll just, that's fine. So I have the normalized counts here. So I'm going to rename them. I have the names here. Okay. So it says rename. So I'll be normalized counts. That's the name. And then we also have this one here. It says we should make it DE results. So I'll say DE results. So these are the files. So the files are now ready. So we can start. So let's go back to the R environment and start the exercise. Okay, so we are in the R environment now. So first of all, let's load our libraries. It's important to load the libraries. So I'll say library, and I'll load the first one, which is gplots. And then I'll load the second one, which is R color brewer. Let's just get the correct name, which is this one. So I will load this one as well. Okay, that has been done. So let's proceed. What we need to do next is to set a working directory. So we are going to do that so that we don't have to specify the full path to the files. So the working directory is where the files are located. That is what I'm going to use. I'm going to set the working directory to where the files are located. So make sure to also do same. So I'll load that here. So I'll set it. So I'll say, let's just say, so to set a working directory, you say set wd, and then you indicate the directory of choice. So for me, I'm going to set it to this directory. So I'll just do it here. So I'll say slash. So this is my working directory. And then I'm going to execute it. Perfect. So the directory has been set. So now it's time to read the inputs. We are going to do that here. So let's start reading the input files. There are 3 input files, by the way. So we are going to load all of them. So we will start now. So I will start with the normalized counts. So I'll say normalized counts read.csv. And then I'll say normalized counts. And then here, even though we said read.csv, the files are tab separated. So we need to indicate the separator because we are reading a tab file, a tab separated file. So we say sep equals, we bring our quote and we say /t. So this will read the file for us. That's for the normalized counts. You can just add that here. So normalized counts, we are reading that. We have done that. Now let's read the other one. That is the D results. So you say, so that will be D results. So the same approach, read.csv. And this time that'll be D underscore results. And it's also a tab separator. So we say sep /t. So let's read this file. That has also been done. Now let's read the genes of interest. So I'll just say Heatmap genes. So read.csv Heatmap genes. So this one too, let's just correct it. I think it's supposed to be this. There's no hash here. I'll redo it again. I just have to get the header, a comment. So I'll say genes of interest. So that'll be Heatmap underscore genes read.csv Heatmap genes, and then a separator, we bring it, that be equals to /t. They should be in quotes. So this is for the genes of interest. So let's read that also. Perfect, that has been done. So now we can proceed. Now, ideally, if you have data sets like this, you can explore. But the focus of this tutorial is to generate a heat map. So I'll do just a little exploration where it's necessary. So take note of that. So let's do it here. Let's just do a quick one before we proceed. So you can check for the column names. You can check some of the entries. So let's just, for example, look at this one. the normalized counts, you can just say column names. Let's just, you can say normalized counts to get the headers. You know, we open them here. Yeah, this one. I think it's this one. Yeah, these are the headers. So that is what we have just done here. And then you can also check maybe first few rows. You can say head normalized counts just to look at some of the rows or some of the records. So we have them here. That has also been done. Anyway, so let's continue. So we will just continue with the next activity. So what we are going to do next is to extract all significant differential express gens. So we are going to use this criteria: adjusted p-value less than 0.01 and log 2fc 0.58. So let's take a look at the instructions from the Galaxy tutorial page. So let's do that here. So this is what we have. So it says extract all significant gens. So we have to use some criteria. That's what I made mention of. So we have that here. It says absolute fold change of 1.5. That's log 2fc of 0.58. So that's one of the criteria. Yeah, so let's just start from here. So to extract all significant gens, we have to use the criteria here. So first one is adjusted p-value less than 0.01, and then absolute fold change greater than 1.5. So i.e. log 2fc of 0.58. So we are going to do everything using the R environment. So we are going to implement this, or we are going to reproduce this using the R environment. So let's go back and then do that. So we are here now. So to do the extraction, we follow these steps. So first, we can display the column names again. Just want to show you something. So DE results. Let me just do it here. Yeah, so this is it. So we are going to extract all significant differential express gens. And we are going to do that using the adjusted p-value. We have that here. There's the column. And we also have the log 2fc, which is this column here. So we are going to write some R commands and then factor in information available in these two columns. That's what we are going to do here. So that's how we do it. So we will say significance gens, and then we say D underscore results. We need to extract the information from that one because it is that table which has that information. So basically we are doing a subsetting here, by the way. So we call the table which has the information we want, and then we say D. Because we have two conditions, we need to place all of them in parentheses. So the first condition is that's the D results table adjusted p-value, this one here, should be less than 0.01. All records that we deem or we say are significant differential express gens they should have adjusted p-values less than 0.01. So that is what we are doing here. And then we add this because both conditions need to be satisfied. And so we have to bring the other one. So the other one is what the absolute value of the result $log2 FC. So that's this one here, log FC. Absolute value should be greater than 0.58. Let me just confirm here. Should it be greater? Yeah, greater. Okay, yeah. So that means this one should be corrected. This comment here should be corrected. So that's it. So these are the criteria we are going to use. Let's just check and make sure that the instructions here are all okay. So we have this one here. Let's just add it. Yeah, okay. So now they're okay. So make sure you type the same thing as I've done here. And also make sure you bring the parentheses where they are supposed to be. It's very, very important. So we have this first condition. We also have this second condition here. So this one, ABS, is used to get the absolute value. So the absolute value for the records in the log FC column for this table, the D results table. That's what we have done here. Now, after doing this, we need to bring a comma because we want to return all the entries, everything from that table, all the entries that satisfy these two requirements or that have their values meeting these requirements or satisfying these conditions. So we want everything. So that's what we have indicated here. So we have the column for this and then this or sets. Okay, so now we can execute this command. Okay, that has been done. So once it's done, you can just do a head or we can just do a count. Let's just count. So we can say, I prefer to use this one because it's a table. I'll do this. So I have 16108. That is what I have. So there's the number of rows, by the way. So it tells you the genes that are significantly expressed. Genes. So that's what I have here. So you can also check for the main one. Which is this one. And here we have 15804. 15,804 genes. And then out of this number, 1,610 of them are significant differential expressed genes. And those are the ones that we obtained here using this command here. So this is how to also subset data using some conditions, which we have stated now. Okay, so let's proceed. So now we are ready to go. So we have extracted the significantly expressed genes. Let's take a look at the next action. Okay, so the overall aim is to get the top DA genes, but we have to first extract all significant genes first, and then we get the top significant genes. That's the next step. So that's what we are going to do next. So now we have all the significant genes obtained. Now we need to also get the top significant genes. So the top significant genes, what we have to do is to get those genes that have the smallest p-values. Those are the top genes. So let's say you want, let's say the 10 genes with the smallest p-values, then those 10 genes become the top genes or the top significant genes. That's basically about it. So just read the information here and then you get the hang of it. So to get the top significant genes, we need to sort by p-value. Here, we have to select the top 20 by p-value. So that means we need to sort based on this column here, p-value, and then we will take the first 20. We are sorting in ascending order. And so when we sort in ascending order, then we take the first 20. Those become our top significant genes. So we have the instructions here. So we have to sort and then in ascending order, and then we get the first 20 rows. Those become the top significant genes. So let's go back and then do the sorting. So we go back to R and then we'll sort. So it says extract the top significant, i.e. the top 10 genes. And this tutorial is 20, so let's just put 20 here. So a 20 with the lowest p-value. So what we need to do is to sort. That will be the first strategy. And then we select the top 20. Or just say select the first 20. That's what we need to do here. So let's start the sorting now. And by the way, I have an entire tutorial on how to sort our tables and data frames. So if you're interested, you can watch that tutorial to get more information about how to sort data in R. So let's proceed. So let's do the sorting. So I'll say significant genes underscore ordered, or I can just say sorted. It's also fine. Any of them is fine. I think I'll just use order. Yeah. And then here I'm going to use the order function to do the sorting. So I'll say significant genes. We are using this table here. And then we bring our square brackets and we indicate the order function. So that will be what? Order. And then in that function, we specify this argument. That will be significant underscore genes dollar p-value. We are sorting on this p-value column. So we are sorting. And with the order function, the default order is ascending. So we are going to leave this as it is. We will add a comma here to return all the entries for us. That's about it. So I think we are good to go. Let me just do this. That's fine. So we indicate the table of interest, the one that we want to sort, and we also indicate the column that we are using to sort. That is p.value. So that's what we have done here. So we can execute this command to get our genes. Okay, that has been done. So now we have sorted, or we have ordered. So we can do this. Let's display the original one, which is this one. So let's say head. I'll bring it here, right? I'll say head significance underscore genes. Let's say the first five rows. So I have them here. Yeah, let's just focus on this one here, the p-value. So look at the order. Now let's do same for this one here. So this is the p-value. So you can see that these ones are in ascending order. It's different from what we had here. Let's check. Are they different? Let's see this. Let's check here. Okay, I think they are all the same. Let's just do it again. Yeah, I think they are all the same. Basically, I think they have been ordered already, so that's fine. So that means we didn't need to do it, but it's fine to do it because you still have to make sure that you are following the right steps. So I think it's fine. Let's just check again. Yeah. Let's just make sure. Okay, so I think basically it's fine. So it's the same thing. You can also manually check, by the way. That's fine. But this is not really important at this time. Okay, so let's continue. So we have our other gens, and now you can select the top 20 gens according to the gallery tutorial instructions. So we have to select the top 20 gens. Let's just recheck. So top 20. So that means we need to get the first 20. So how do we get the first 20? Which will be our top gens. So I'll bring that here. So that's going to be, I'll just say top 20. Let's say top 20 gens. I think this is much more descriptive. So top 20 gens is going to be the first 20. So that will be significant gens ordered. We are using this one here. And then we bring a square bracket and we say 1 to 20. That is what we have here, 1 to 20. And then outside the square bracket, we will say dollar symbol. So why are we saying symbol? We just want the names. Okay, we don't want everything, we just want the names because we are going to use the names to do a subsetting later on. Now let's take a look at the table of this one here. Let's check the D results. So we did a subsetting to get, let's just check, I think it should be somewhere here. Yeah, so we did a subsetting to get the gens with the smallest p-values. And so what we want is to, instead of getting everything here, now what we want is just the gen names, which is this one here, this column here. So that is why I am using dollar symbol. I just want the names. Don't worry, I'm going to explain everything later on. But for now, we just want the names because we'll use that to extract the data that we are going to use for the heat map. So for now, we just want the names. So that is why I'm using this command here. Okay, so first 20, and I want just the symbol column, the entries. That's for that one. Remember, we had this column. We had the symbols here. Let's just take a look at some of them before we proceed. I think I had them somewhere here. Okay, I think I should, yeah. For example, this symbol. So we just want the symbols here. That's all that we want. Okay. You can also try with other columns as fine. You just need to make sure that you are using the right column of interest. Okay, so this is what I have. So I'm going to execute this command now to get the top 20 gens. Just the gen symbols. So I've obtained that. So now I can just say top underscore 20 gens to display them for me. So these are the gens. That's what I have here. Okay, so I need this information. So I now have it. So after getting this information, then what we need to do is to extract the normalized counts for these gens. Now we have two tables. Okay, so we have this one here, which has the differential expression analysis results. So what we did was to use the information here to get the gens that's of interest to us, those ones that are top gens. We want the names. But what we actually want to plot, what we are going to use to generate the heat maps is the normalized count, which is this one here. But here, we have lots of gens, over 15,000 of them. So we only want just a subset. So the subset is the top 20 genes. So what we got is the gene names. The gene names which I showed here. And so the gene names, once we have the gene names, then it becomes easier to now come to this table, the gene counts table here. And then because we have the same symbols here, we can now use those symbols to extract the associated counts. That's basically the rationale behind whatever we are doing. So we have the names. So we just come and then pick it. So just consider the names, or let me just say the symbols as what? IDs that we can now use to extract other information. Okay. So let's say if it's a database, you have your student ID or whatever, or staff ID. Then once you put in the staff ID, you can get information about you. It's all linked. So this time we are going to use the gene names, or let me just say the gene symbols, to come to this table, which we'll do in R, by the way, and then extract the normalized counts that we want for the heat maps. So that's straightforward. So let's get back to the Bioinformatics tutorial page here. So it's the same thing. It's selects that, and then we can now extract the normalized count for the top gens. The normalized count for the top gens. So that's what we are going to do now. So we even have the list here. Let's just compare to see. So we can do that here. Okay, so I'm just showing the names that we have. I think you can try it and see. So let's just say we have one of them, XKR4, which can be found here. Let's check. It's supposed to be somewhere here. We also have SOX17, etc. So we have all of them here. Okay. Anyway, so these are the normalized counts, by the way, for all of them, but we are doing a subset of that. Okay, so we can proceed now. So when you do the extraction, we are going to get something like this. Yeah, so this will be the final output we'll get once we do extract. So let's just do that in R. I think it will be better if we do it in R so that you get a hang of it. So we have the next step here. So what we are going to do next is to extract the normalized count for the top gens. So it's getting interesting now. So here we have to do some steps. Okay, so if you're extracting, it's also easier to add some additional information. Let me just say do further processing or further preparation of the files. So how do we do this? So what we need to do is to supply the information that we want, and then I'll return to us. So to extract a normalized count for the top gens, we'll say final data. That's what I'll call it, because that's the final data we'll use. And I'll say normalized count. And then I bring my square brackets again. And then in the square bracket, I'll indicate this command. I'll say normalized counts $symbol. And I say percentage in percentage. And we add the list, which is top 20 gens, and we bring our comma. So this is the final command. So what is happening is that we indicate the table that we want to extract the information from, which is this one. And then we indicate some conditions here. So here, this one indicates that for that table, this column here, all entries in that column, if they are found in this list, that's the top 20 gens, then they should be returned to us. That's basically what we are doing here. So for the top 20 gens, we have them here. And so what is happening is that because we want just information for only those ones, i.e. the normalized counts, if you go to the table where we are doing the extraction from, take a look at one column, which is the symbol column. And then for the symbol column, all entries there, if they are found in the top 20 genes, return them. Now let's take a look at some here. So there's the gene symbol. Okay, the gene symbol, and then that is what we extracted. That is this one here. But this gene symbol, which is found in the differential expression analysis results file, can also be found in the normalized count file, so which is here. Let's just go back, which is this one here. So basically what we are doing is that the command will take a look at the normalized count file and then take a look at the column, which is symbol column, and then all entries in that column, if they are found in our lists, our top 20 list here, then all records, or let me just say the normalized counts information for those genes should return to us. So that is what this one is for. So this one here, that's so that all the records that can be found in this one should be returned to us. And then the comma here also adds up to give us everything, because we want all the columns, everything. We want all the records to be returned to us. That's basically what we are doing here. Okay, so now we can proceed. So that is the rationale behind this command. So don't be scared, don't be afraid of this one here. It's just a simple instruction. So now we can proceed. Now that we are clear, we can proceed to do the extraction. So let's execute this command. Okay, so it has been done. So now we have our final data. So the final data, you can say head final data, then we get them here. So final data, you can also say dim final data. We should expect 20. That's what we have here. We can also get the call names. And we have them here. So this is the counts data, the normalized counts data. We have them here. So this is what we need. Of course, you can also do a head, which we did earlier on, by the way, to get the counts here. So we are almost there. Now, with what we have, with the heat maps, we need to also add some additional information. We need to get an index. We need to set an index for it. Now, this is the current index. Let's say row dot names. Let's say final data. We have some numbers here. These are numbers. But with the heat map, we need this information. Let me just show you. We need this. Let's just take a look at this one. So we need this and we need this. So that is what we need to get. So at the moment, we have the information for this already in the data, but for this one, we need to set it. So that's what we are going to do next. So these ones, that's what I showed there. But for the other one, which is this, the gene names here, or the gene symbols, that one, we have to do some further processing on data to get that. So of course, they can be found in this column here, but this is not where they are supposed to be. They are supposed to be the row names rather. So we are going to change the row names and then use the symbols, the gene symbols. And then we are also going to remove some unwanted columns. So we are going to first set the row names to this one here, and then we are going to remove all the other columns as well. So we have these 3 columns. We are going to remove them, and then we will get there. final data itself. So we are still going to process the final data here. So that's what you need to know. So even though it's final, we are now going to make it completely final. Let me just put it that way. So let's start the exercise now. Okay, so let's do that. So that's what we are going to do. We will set the index. So we will say to set the index for an R table or data frame, we say row.names and we say final data. We'll say it's equals to final data dollar symbol. So let's set it. So now if you say row.names for final data, we should see the gen symbols. That's what we have here. So now we can proceed. So that's what we are going to do next. We have set it, but then we still have some unwanted columns we have to remove. If you do this column names, we did it earlier on. We are going to find these columns. We have to remove them. So what we are going to do next is to process the final data here again. At this time, what we are going to do is to remove these columns from that data. So that's what we are going to do next here. So let's proceed. There are several ways to remove columns, but I'll cover them in details in other tutorials. So for now, let's just use this command here to remove the unwanted columns. So how do we do that? So we will say final data, and then we say final data again. And then we bring our square bracket. So here we are going to do this by indexing. Okay, so that means we will just use the numbers. So we have 1, 2, 3. That's basically what we are doing here. So we say minus C, and then we say 1 is to 3. That's 1 colon 3. That's it. So this is the command. So here, what we are doing, that it's still a subsetting command, but this time what we are doing, that we are rather removing. That's why the negative sign is there. So that C, that means we are removing. So first to third, 1, 2, 3. That's what this will do for us. So let's execute this command. That has been done. So now if I say column names final data, you will find that it's not there. I mean, those columns are no longer there. Okay, we can still do our row.names to just confirm. Perfect. So now we can do a head final data. Let's say first 3. We have them there. So this is the final data itself. This is the final. So finals of finals. So now we can proceed. We can now generate the heat map. But before we generate the heat map, we need to convert our data to a matrix. That's the requirement for using the heat map too. So how do we do that? So let's just take a look at our final data here. It's a data frame. Okay, so to convert a data frame to a matrix, that's how we do it. We are going to do that here. So there's a data frame, so we are going to convert to a matrix. So to do that, I'll say final data, because I want to assign the new changes to the same variable. So that'll be data.matrix. Now I'll say final data. So this is how we convert a data frame to a matrix. So let's execute this command now. Let's just make sure everything is okay, and then you can execute it. So that has been done. So now if I say class final data is what? A matrix array or matrix array. So now we can now generate the heat map. So let's generate the heat map now. All right, so let's do that. So we are going to use Heatmap 2. So that's what we are going to do next. So to generate the heat map, you need to make sure that you have followed all the instructions here to get your final data, which is a matrix, and then you will call Heatmap 2. But before we do that, let's take a look at the instructions that the Gala tutorial used to generate the heat map. So let's go there. So here I want us to take a look at some of them. So it says enable data clustering. No, we will need to indicate that, or we will need to specify that. And then labeling columns and rows, that one is already done. The package will take care of it. And then coloring groups, blue to white to red, we will also indicate that. And then we also have data scaling. Okay, so scale my data by row. We are also going to do that. So that's about it. So we need to specify all those stuffs. Okay, so let's get back to the R environment and then start the exercise. Or let's just say continue the exercise. Okay, so we will start with this one here. So we are going to generate the heat map. So I'll say heatmap.to. Now I'll say final data. Because we don't want clustering, I will say vin equals false, or vin equals false, and then dendrogram equals none, and then scale equals row. So this is how we do it. So let's execute the command. Okay, the heat map has been generated. Now if you take a look at what we have here and you compare to the one here on the Gala tutorial page, there are two things we need to modify. One is the color scheme, and then two, trace lines. So if you take a look at our heat map, you see these lines. These are trace lines. So we need to disable that. So these are the two things we are going to do now. So let's do this. Let's say disable trace lines. So to do that, it's still the same command. We'll use the same command here. And this time we'll say trace equals none. So that will disable the trace line. So let's do that now. Nice. So now the trace lines have been disabled. So that's how we disable trace lines when using Heatmap 2 to generate your heat maps. Let's add one more thing. So what we need to do now is to use the same color scheme as this one here. So let's get back. So we are now going to change the colors. So we are going to change the color. And that is why this library comes into play. Okay, that's why we need it. We need that library. So we are going to change the colors. I think I already have it here. I'll just do this. Yeah. So we are going to change the colors in the heat map. So to do that, I need to define a color. So I'll do that here. So I'll say colors. You can use any name of your choice. I'll use this one, and I'll say color ramp palettes. That's what I'm going to use. Let me retype it again. Yeah, this one here. That's what I'm going to use. So I'm going to set the colors to blue, white, red. You know, on the Tutorial page, we have that here. It says blue, white, red. So I'll use the same color scheme here. So to do that, I'll say C, and I'll say blue, and I'll say white, I'll say red. In some tutorials, instead of single quotes, you see double quotes. So it's still the same thing. So where you see double quotes in some tutorials instead of single quotes, it's still the same thing. So don't be worried or be bothered about me using single quotes here. I can still use double quotes. That's fine. Okay, so I have defined the colors, or let's say the color scheme. So that's what I'm going to do next. I'll just execute the command, and then I'm now going to add that information to the Heatmap 2 command. I'll just do that here. Let me play this and do it. So I'll do that here. So I'll say heatmap.2, or let me just do a copy and paste because I already have the command here. I'll just paste it here, and then I'm going to define the color. I'm going to specify that here. So to do that, I can put it anywhere. Let me just put it here. That's fine. So I'll say col equals colors, because I specify that here. So I'll execute this command, and that will give me a nice and cool heat map. So this is what we have been waiting for, and now we have it. So there's the heat map. Now with this heat map, if you compare with what is on the tutorial page, maybe the order might be different for this and this, but it's still the same. Let's just check. The order for the genes may be different, but it's still the same data. So don't be bothered if you see that your gene is here or here, and then on the Galaxy tutorial page, the gene is in another place, but it's still the same. So what I want you to focus on is the colors, okay, and then comparing them. So for the genes, it doesn't matter where they are, they will still have the same color intensity because they are being compared to each other. So that's what you need to know. Alternatively, you can arrange your genes also so that they are in the same order as what is on the Galaxy tutorial page here. And once you do that, you'll be able to get the same color arrangement here. But that's not anything to worry about because it's basically the same. We have the same information being displayed for us. So that's the only thing that you might notice when you compare your heat map to this one here, but it's fine. So that is it. Okay, good. So now we have generated our heat map, which is very, very great. So we use the top 20 genes. Now, what about if you have custom genes? Maybe instead of top 20 significant genes, you want to use your own genes. Maybe you have some genes of interest, genes that you are interested in, and you want to generate a heat map for that. It's possible to do that. So that is the next part of the tutorial. So we are going to do that also now. I think it's important we factor that in. Now, let's take a look at our data, the genes of interest. So if you take a look at our data, we have heatmap genes. So we have that here. I think I open it in the spreadsheet. I will do that. I had that here. Yeah, so these are the genes of interest. So what we are going to do, we are going to extract the normalized count for these genes, Then we are going to use them. We have, I think we have about, let's say, 31 or so. I think 1, 2, 3, 4, 5. Yeah, we have some about 30 or so of them here. So it's possible to also extract the information and then use that to generate the heat map. So let's try that. Let's go to the R interface and then do that. We have already loaded our gens of interest. Let's just say, let's check the names. I think I use heatmap gens. Yeah, this one here. Okay, so I'll just do this. Let's see. Let's try this. So we have 31. Okay, so I can say heatmap gens. So I have them here. So we are going to extract a normalized count for these gens and then use them. So it's basically the same rationale that we use for the other one. So we are going to follow a similar approach. So how do we do that? Let's proceed. So let's do this. Let's say gens of interest. Gens of interest. And we indicate the normalized counts data. So that is normalized counts. And then we bring our square brackets and we say normalize count dollar symbol. Then we add this command to it and we say heatmap gens dollar gen ID. We bring our comma. So this time, this is the list that we are interested in. So we want the gen ID column for the heatmap gens data. Okay, so that's what we have here. So once we have this information, we can now execute the command to get our gens of interest. So let's do that now. Perfect, that has been done. So now we can confirm, we can check, we can take a look at what we have. So we can say then gens of interest. So we should get 31. So that's what we have here. Okay, so everything is now going as we planned. Everything is now going on very smoothly. So now we have the gens of interest. So what do we do next? We have to set the index again. So here we will still use the row.names. So when we come here, we say row.names. That is gens of interest. And then there will be what? Gens of interest. And then we say dollar symbol. Yeah, dollar symbol. So we should have the same thing. I repeat, we should have the same thing there. Okay, so that's the index. So we are setting it now. Perfect, that has been done. So you can confirm by saying row.names gens of interest. So that should give you your gens of interest. Okay, that has been done. So now we can proceed. So what we are going to do next is to delete the unwanted columns. So we have to remove them as well. So for the unwanted columns, let's just list again, call names gens of interest. We are going to move these 3 columns. So let's do that now. So To do that, we know the drill already. So we will say genes of interest, and then we specify again, and then we do our subsetting. So we have -C, 1, 3. So this becomes our final command to remove the columns that we don't want. So let's execute this command. That has been done. So you can confirm by checking your columns again, and they should have this one here. So we are ready to go. So now we are going to convert our data frame into a matrix. So we say genes of interest, and then we say data.matrix genes of interest. That has been done. We can confirm by saying class genes of interest. So that should give you a matrix. So now we are ready to generate the heat map. So we can just do it straight out. So with this one, I'll just do a copy and paste of the command that we use. This one, because it's basically the same command. So I'll just paste it here. And then when I come here, this is the data. So I'll just change this to genes of interest. That's basically it. So that's the only thing we need to change here. So we can execute this command now. Perfect. So that should give us something like this. Okay, the heat map has been generated. Now let's take a look at something. So if you compare this to what we have here on the Galaxy Tutorial page here, let's just check that. We see that for the Galaxy Tutorial page, we have the genes on this side, and then we have the conditions here. Of course, here they rename the conditions to these ones, but we are not doing that. But what I want to highlight is that here the genes were here. They were on this section. Those were rather the columns, and then the conditions were rather the rows here. And so it's possible to do that. So all that I have to do is to swap that. So it's like what we have here, and then we do a swapping. So what is here, these rows here become the columns, and these columns become the rows. So basically we'll swap the genes and then the conditions. So it's possible to do that. So we will do that so that we can get something similar to what we have done here. At the moment, what we got is still in this format, but we want it to be in this format, in this particular format here. So we can do that as well. So let's go back to the R environment and then proceed. By the way, here too, we had 31 genes, but you can see that it's just a few of them because of this setup here. So you can just pull your widget up and you get the list, or you can also save to an output file, and that should also help. But anyway, let's just do that. So we are just going to swap the rows and columns, and so we'll do that here. Let's just check. Okay, the heat map has been generated, so we have them here. Now you will notice something here. So you will notice that we have 31 genes, but here we have less than 31 genes being shown or displayed. So it's because of the arrangements here. So you can drag your widgets up a bit and that will display the entire list. Alternatively, you can save the plots to a file, to an output file, and you can have all the lists being displayed for you. That's even better. Now let's take a look at something else. So if you compare this to the gallery tutorial page for the custom genes or the genes of interest, you see that the genes of interest were here. This section, this side, and then the conditions are on this side. Of course, here they rename the conditions, but I will leave it as this. It's possible to do that, so you can try your own. If you have challenges, you can also contact me, but I am focusing on this one. So we can also swap. So instead of having the genes here, we can bring them to this side. We can make them the columns rather. So it's possible to do that here. So what we are going to do is to regenerate the heat map, and this time we will let the genes be here. The genes will rather be the columns, and then the conditions will be the rows. That's what we are going to do next. So it's possible to do that. So let's go and then do that now. So let's check. Okay, the heat map has been generated. Now, if we take a look at what we have here, we use 31 genes. So let me just say the genes of interest were 31. That's the number. But here, less than 31 is being displayed. This is because of the setup here. So you can just drag your widgets up a bit, and this will display everything for you. Alternatively, you can save this plus to an output file, and then you can view using your image viewer. So that's it. Now let's compare what we have to the one on the gallery tutorial page for the custom genes or the genes of interest. So let's scroll down a bit. So when you scroll down, you will see it here. That's what you have here. Now let's take a look at something else. If you compare what we had to what is here, you'll see that the conditions are rather the rows for the gallery tutorial page, and then the genes are the columns. But ours is the other way. We have the genes as rows and then the conditions as columns. That's what we have here. So we have to change it. Okay, we want to get the same figure as what was obtained here. So that's what you are going to know. So it's possible to do that. So all that you have to do is to do the swapping. So the swapping, I will show you just one trick, which is very quick and simple. So with the swapping, what we have to do is to transpose our matrix, the matrix that we use. So when you transpose, that means you are swapping the rows and the columns. But you will do that here. It's just a one-liner command. So we will do that here. So we are just going to copy this same command, and then when we come here, the data, let's just clear it. I'll just do it here. So I'll clear this. So what I'm going to do, I'm going to transpose and then place it there. So I'll say T, and then I'll say genes of interest. So I have transposed it. Okay, so once I do that and I execute the command, I'll get it here like this. So now if you compare what we have here to the one on the gallery tutorial page, they are now similar. So this is how we do it. So everything is good now. With the transpose, you can also create another command separately, get the transpose data, and then supply that into Heatmap 2. That's also up to you, but I just thought I'll show you this simple approach. So that's how we do it. So that's how we generate Heatmap using Heatmap2 function in R, or let me just say in the gplots package. Okay, so that's how we do it. So I have other tutorials on how to generate heat maps. So check the description box for that playlist, and you can also check the playlist here to show you how to analyze RNA Seq data.